Hi guys, Stubborn Gamer is here. So today we are playing Cell Lab. So first I'm going to do experiments. And if you don't know about this game, so in this game we just make a cell and we loot it. So guys, sit back and enjoy and subscribe my channel and like this video. So first we are doing contaminate with random cells. Let's do everything 100. Yes. Whoa. So we have done everything 100, guys. So let's start. Oh no, guys. Let's name our sample. Hello. Let's microscope. So guys, I don't know what to do in this plate, what to do in this game, so I'm going to do tutorial first. Okay. Okay. What you see here is a petri dish we have prepared for you. To zoom with this microscope, do an inverse pinch with 2 fingers. Please increase the magnification by a factor 4. Great. Now move the substrate so that we are looking at the brighter area at the top of the petri dish where sunlight is visible. Zoom so that this area covers this screen. Swipe with 1 finger to... Great. Now move the substrate so that we are looking at the brighter area at the top of the petri dish. Zoom so that this area covers this screen. Okay. I see you have used a microscope before. This microscope is equipped with a micromanipulator. One of its uses is to insert cells from a genetics lab on the substrate. Can you insert one of our recently developed cell in the area with sunlight? Tap where you want to insert cell. Okay. temperature, not too hot. A microscope also has a system for controlling the temperature of the substrate. Turn up the temperature to observe setting where the cell live but move slowly. The temperature setting is chosen by the selector in the top right. Oh. Cell tracking. Now you should have a lot of cells. This makes it hard to keep track of them. But luckily the microscope has a visual tracker. The tracker overlays a marker on the selector cell and also move the substrate to keep it in view. Set temperature to observe again and tap on a cell to tell the microscope to track. DNA extraction. Good. Sometimes we bring in cells from nature or nurses to study. We can then extract their DNA using the microPT to study it in the genome editor. Let's do that on one of the cells on the substrate right now. We put a mutagen in the substrate, so some cells should have mutated. Pick an interesting one and tell the tracker to follow it. Freeze the substrate before doing it. Sometimes we bring in cells from nature. Okay. Good job. Next step is to go to Genome Editor, tab Genome and press load. There choose load from microscope and the microPT will automatically extract the DNA of the cell in the tracker, sequence it and send it in digitized from to the Genome Editor. Great, now we have the genome of the mutated cell in the Genome Editor. Your last task is to sterilize the substrate with the built-in autoclave to prepare for the next experiment. This is done using the reset button. Good luck and keep your fingers out of the autoclave. Reset. Great, now we have the name of the Guys, I'm not able to understand that what they are saying. Great, now we have the genome of the mutated cell in the genome editor. Your last task is to sterilize the substrate with the built-in autoclave to prepare built-in autoclave. So guys, we are leaving this here and we are doing LA. So let's do challenge. Use the genome editor to make a species that thrives in the environment of this challenge. When ready, put a cell on the substrate under the microscope and unfreeze it to see if it is a variable of 100 cells are needed to complete this challenge. So guys, we have to put 100 cells. The default cell width will suffice for this challenge. All cells need energy and the okay. Oh, I got this, guys. We are making cell, guys. Guys, we just completed the challenge. Oh, we got this. So we are doing LG3. Inserted cells. Guys, I'm not getting it. So let's do an experiment on our own. So let's do it. So guys, I found this game very interesting game and a knowledgeable game. It is very good for kids who are studying science and that's all. It is a very knowledgeable game. So I like it a lot. I think so that we will make second video of this also. I don't know when. So guys, this was all for today. Hope you enjoyed this video. Like and subscribe my channel. Bye.